Okay, so, gonna yammer for a while and see how far we get, and then we'll do lab at 3, and we'll, meet in Brim and jump in the truck and go to my house where there are not puppies anymore. And I haven't actually done the walk that we're gonna take, so I'm not sure.
If it's like completely muddied.
Yeah, it'll be pretty wet out there, and,
I'm just excited to know where you live.
yeah.
Is it supposed to like rain a lot more? Because the traffic might.
Not really. Not till
tonight
maybe
it's
tomorrow.
Tonight's gonna rain some, and then like Saturday and Sunday it's supposed to rain. And, but we got 3 inches at my house last night, so.
Yeah.
So, that was so not stressful with puppies. Anyway, okay. So we need to finish up the ecological stoichiometry, and then a couple other things with nutrient use, and then we'll do something totally different after that. But you saw this whole slide, right, last time?
Maybe the one before it.
Maybe the one before, okay.
Talk about.
Oh, yes, we didn't actually get to it.
Yeah.
Okay. Okay, so ecological stoichiometry. I'll just put it all up there. So, so you've done stoichiometry in chemistry class where you're like balancing equations, right? That sounds familiar. So it's kind of like that, but we're not really looking at chemical reactions. We're just looking at like ratios of elements in various organisms. And so ecological stoichiometry, looking at the elemental needs of organisms, and then kind of the impact on food webs and ecosystem function. So, for example, if a fish converts 10% of the carbon that it eats into biomass, and the rest is used for, that E means energy, so the rest is used for just surviving life as a fish or maintaining life as a fish. And so the like kind of required elements that it needs might be 100,060 carbons to 16 nitrogen atoms to 1 phosphorus atom. But then the food it eats is around 106 to 16 to 1. Remember the Renfield ratio from last week? Yeah, so that's where that's from. So therefore, we've got a bunch of extra nitrogen and phosphorus that the organism, that fish is eating, and it has to get rid of it. So, so it's eating extra N and P to get enough carbon, and then it will excrete out the extra nitrogen and phosphorus. So urine or uric acid has a lot of nitrogen compounds in it, getting rid of nitrogen, so that's a pretty common thing. And so some people get really into stoichiometry and looking at the ratios of elements in food resources, and then what it looks like in the consumers, kind of up through the food web, and trying to figure out what elements are going to be limiting. And so different species have different ratios. That 106 to 16 to 1 is like an average. The Renfield ratio is an average across a bunch of organisms. But some examples from our aquatic systems. I guess we haven't seen Daphnia. Did Dr. Grubaugh talk about Daphnia? Water fleas. Okay, they're so Daphnia and copepods are both little tiny crustaceans, and they swim around and eat algae. And so they have very similar lifestyles, but Daphnia just needs more phosphorus than copepods do. So, so they're more likely to be limited by phosphorus than copepods. So if phosphorus is kind of low in a system, copepods might do a little bit better. And so kind of knowing what's going on with the stoichiometry of different organisms, that can tell you something about what might limit them, like what elements might limit them. Another example, crayfish need more phosphorus than snails do. So similar kind of thing. Does that make sense, kind of, sort of? So that's a very quick look at ecological stoichiometry, and probably do a whole semester course on that. But I would like to discuss next something different called the paradox of the plankton. This idea was developed by our old friend G. Evelyn Hutchinson, 1961. And it's kind of combining Liebig's law of the minimum. Remember Liebig's law of the minimum? So whatever element is in shortest supply relative to the needs, that will be what's holding back growth. And so a combination of Liebig's law and the competitive exclusion principle. Who remembers the competitive exclusion principle? From general ecology, maybe? Anyone? Bueller?
So where two organisms compete with each other and then one just eventually outcompetes the other?
Yeah. So if we look at like ecological niche, so all the resources that organisms need, if there's any kind of like overlap between two organisms, they're competing with each other, and generally if there's enough overlap, one of them will outcompete the other and like completely displace one of them. I guess to look at it in kind of a more extreme way, you had like two organisms that one whose niche was completely inside of another one's niche, that one's gonna outcompete the one on the inside. It's got a little bit more of a niche. And so basically, competitive exclusion principle, if you've got organisms that basically have the same needs, they have the same niche, the same needs, one of them will outcompete the other. So you don't expect to see organisms that need exactly the same things living together in the same place. So thinking about those two things, Hutchinson was asking, why are there so many species in the phytoplankton? So, like, imagine you're in the plankton, you're floating in the water. We're assuming the water, with this question, we're kind of assuming the water is fairly uniform with nutrient availability and that kind of stuff. You've got all these different species floating around, and they're all kind of using the same resources, we think. So why hasn't one species like outcompeted all the other ones? Like, why do we have a lot of species in the plankton?
Just too much space.
We need too much space.
Maybe
That would be the limiting factor though, if there's enough space.
maybe predator control.
Mm-hmm.
Not able to outcompete the others because predators coming in and
Yep, that could absolutely be. We'll make a list of possible things. So predation could be why. There could be some species that get preyed upon, not predated upon, preyed upon more frequently than others. So that might kind of shift the balance of competition for sure. Don't forget about viruses. So viruses can negatively impact certain species but not others, because viruses are very species specific, even those microbes, even if we don't know what the viruses are. Also, I think this is probably a really big one, is it's kind of not necessarily correct to assume that throughout that water column that everything's the same. There's probably little areas that have like a little bit more nutrients, and if you've got a copepod swimming around and a copepod peeing, and then that area where there was copepod pee, that's a nutrient-rich area that an algal cell might enjoy, for example. So there's likely these little tiny patches throughout the water column of more nutrients, and so it's not really a uniform.
like when you go swimming and all of a sudden you feel a warm spot in the pool.
Exactly. Exactly. Good times, good times. There could be mutualisms where two species together kind of, I don't want to say help each other out because it sounds too swell, but the presence of the other one benefits one species. So that could be part of it. And then it may just be a matter of time scale issues. So the like seasonal changes in a lake with all the turnover and everything that we talked about, the time it might take for a lake to sort of reach an equilibrium of it's not really changing that much, may be much less than the time it would take for organisms to outcompete each other. So the lake might just change faster than all that competition. It might take less time for the lake to change than for the algae to outcompete each other. And so by the time the lake changes, then the competition is different. Alright, that makes sense? More or less? Okay, a few other fun nutrient things that aquatic ecologists like to talk about. One is nutrient subsidies, where nutrients that are from outside the system get delivered into the system in some way. Excuse me. So for example, some rivers have marine-derived nutrients, and salmon returning to spawn is like the big one here. So you've got, so salmon, they live their life in the ocean or the Great Lakes, and they go up a river to lay eggs and die. And so they lay eggs or fertilize eggs and then they die. And those little eggs hatch, and those little baby salmon might live in the river for a while, but eventually they'll go out to the ocean to live their life. And then when it's time for them to spawn, they'll come back and then they'll die. And so that all those carcasses that end up in the rivers, those there's a lot of nutrients there, and so you end up with a lot of dead salmon. They're decomposing, and that delivers a bunch of nutrients to the stream. But all those nutrients came from the ocean because the salmon were growing in the ocean, and they got all their nutrients from the ocean, and then they came back up their stream and deposited those nutrients there. So, so that's a nutrient subsidy. The fish coming back are providing nutrients for that river that otherwise wouldn't be there. We also have terrestrial subsidies to streams in particular. So allochthonous inputs, what are those again?
Outside sources.
Yeah, like what's an example of an allochthonous input to a stream? Yeah, so like leaf litter falling, so overhead trees covering the stream, they'll drop their leaves. Those leaves are going to be a source of nutrients for that stream as well. I think that's, extra stuff. Yeah, that's finally everything for the nutrient junk. Any burning questions? PowerPoint has a few different things in it. But we'll talk a little bit about evolution and biodiversity and pollutants and just a bunch of random things. So, when we think about evolutionary change and diversity, so evolution basically results in diversity. So the more time there is for evolution to happen, the more diversity we can see in aquatic ecosystems. And this is a combination of time, so the more time that you have, the more opportunities for new species to evolve. Also, reproductive isolation is really important for the development of new species. If you took evolution, remember that, talk about that. So basically, if you've got two populations, so in 130, I like to talk about the squirrel population in Martin compared to the squirrel population in Kenton. Kenton squirrels have the albinism gene, you get white squirrels down there, and if the Martin squirrels and the Kenton squirrels never breed with each other, the Martin squirrels are probably not going to develop that albino gene. But if they were to breed with them, they might get that. And so the fact that they're reproductively isolated means that any differences that they happen to get from mutations that just happen, and if those mutations stick around, they'll just keep adding up until you've got two different species. So if you can keep populations separated from each other, and if new characteristics show up and they keep accumulating, they'll become different enough to be different species. So in freshwater ecosystems, if you have a really long time, and in particular lakes are isolated from other lakes frequently, then we can get some really interesting evolutionary change. And this graph here is showing some interesting diversity numbers for some of these really ancient lakes. And I mentioned these ancient lakes earlier in the semester. So Lake Baikal is our oldest lake at like 20 million to 30 million years old. Lake Tanganyika in Malawi, I think those were like 1 to 2 million years old. Lake Victoria, also in Africa. Lake Ohrid in Macedonia. And the Great Lakes are about 10,000 years old. But this is showing some endemic species. So teleosts are fish, mollusks, and crustaceans. And in the older lakes, we see a lot more endemic species, so we only find them in those locations. Lake Baikal has a lot of endemic crustaceans, for example. Or some other endemic things. I think these are mollusks. Lake Baikal, they even have like their own seal. Like they've got endemic diatoms also. So they have a lot of unique species that you don't find other places. So a lot of these really old lakes, we get high diversity. And the Great Lakes are bigger, but they're younger. The Great Lakes are overall bigger than a lot of these lakes, but since they're only about 10,000 years old, we don't have as much endemism in them, and so most of these species are more cosmopolitan. Cosmopolitan just means they're found all over. So some of these really old lakes and rivers are going to have some interesting species. And like the Amazon River, they've got a whole bunch of their own species there as well. Alright. I want to talk about groundwater ecosystems because no one really talks about that. And so in groundwater, like if there's enough space to get like little channels of water that can move, some space for organisms to move, you can get animal life that live underground. So that's kind of weird. And these are are some groundwater organisms. Notice any characteristic that the four of these share?
Transparent almost.
Yeah, transparent almost.
Kind of like antennas, or similar to crustaceans.
What about antennas?
They say they kind of look like they have antennas.
Oh, yeah. These, so these are crustaceans. They do have antennas. So are these.
They're all
These
blind.
are insects. Yeah, they're blind and they're colorless. So they've lost the need for eyes, so those traits are kind of gone, and they haven't developed any color because no one can see them anyway. But they probably have really good chemical reception and sensory perception also. So we don't know a whole lot about, we know much less about groundwater ecosystems than above ground ecosystems for sure, but there's there's high endemism in groundwater ecosystems. We find things there we don't find any other place. And some of these organisms are obligate groundwater organisms, and they're referred to as stegofauna. And like all y'all just said, there's no pigmentation in these dudes, no eyes frequently, and larger sensory appendages. They also might have really slow metabolism and just their life moves very slowly. Groundwater tends to be cooler, so that's going to slow things down also. Groundwater temperature is often like the average annual temperature of an area. So like spring-fed rivers in Florida are around 70 degrees. Spring-fed rivers in Michigan are like 50 degrees. So, which is a good one. So that's just kind of weird. What else are we going to talk about? A little bit about genetic diversity. So this is really an important part of species diversity. So let's say you've got a species and it becomes endangered or threatened. You've got a population that's gotten really, really small. What do you think happens to the genetic diversity of populations that get really, really small? They go in the crapper. So can we get that genetic diversity back?
Maybe.
Not without tampering.
Yeah, not without giving it back to him. Not without a really, 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 really long time, like millions of years. Once genetic diversity is gone, we can't get it back. So even if species recover, like the population size gets larger, and so we have a lot of numbers, we may have lost a lot of genetic diversity in those species. So genetic diversity also really important part of species diversity. And thinking about like long-term species survival. So the kind of a benefit of, one of the many benefits of genetic diversity is if the environment changes, there's enough different genes and alleles out there that you could have individuals that are surviving that. If you don't have a lot of genetic diversity and the environment changes, that might kill off everybody. So long-term survival in changing environments, we need genetic diversity. And it's interesting, so a lot of the aquatic people are ecological types, and they're ecological types because they're more interested in like the biology 130 stuff like ecology and evolution, and less interested in the genetics stuff. But we've started to get people that are looking more at genetics of our organisms, and so some ways that we are kind of like adding molecular biology to ecology is using DNA sequencing to get a handle on diversity of things that we have a hard time telling apart. So microbes like bacteria that all look the same in the microscope, we can look at their DNA sequences and figure out maybe how many species we have. And so that's been a thing recently. Have you heard of DNA barcoding? So, one of the things that some aquatic ecologists like to do is go get like an algae sample or a macroinvertebrate sample and see all, like try to find all the species that you can, find out what species you have there. And it's kind of a pain in the ass. So, DNA barcoding is when you've figured out like segments of DNA that you can look for in a sample, and if it's present, you say, okay, I know that that species is here. So in theory, you can have an environmental sample and send it off to Magical Molecular Lab, and they come back with a list of what you had in there. You don't have to sit there and look at everything. But that's still in the developmental stages. It's not anything that we can really use instead of looking at stuff. But yeah,
Is that a scannable barcode?
that's just a joke there. Yeah.
No, like, I
Barcode.
wanna scan it to see what it pops up.
Oh, I have no idea what that is. I just Google barcode and Google image search.
Might scan it.
Get your Walmart scanner out and see what it is. I don't know. Could be nothing.
Most of them like these.
Could be a magazine. Could be could be chocolate chip cookies. Have you heard of environmental DNA or eDNA? Yeah, figured you had. Environmental DNA. This is kind of cool. Where you can take an environmental sample. Let's say you're looking for a certain organism. You can take an environmental sample and look for the DNA of like rare organisms. So if you're like, I wonder if the endangered, I don't know, purple polka dotted snake lives in this area. And if you have DNA information for that, you can just take samples and look for that DNA. And so just an organism like moving through an area, they might shed some scales or skin cells, or they poop or they pee and leave behind some of their DNA. And so even if you don't see the organism, or you don't, like you're not trapping them or looking, like you can't find it, but you can tell if it's there by looking at eDNA.
Oh, like based off of that one movie where a guy was trying to protect the albino bears or some kind of like spirit bear. It's something similar to what he did. He could never really prove it except with a tuft of fur.
Yeah, there you go. So yeah, it's basically like a CSI looking for DNA left behind by a rare organism.
I guess it depends on, because a lot of that like, I guess spoils quickly, depending on like what you collect.
Yeah, I don't know how long that stuff can stick around.
Because I'm like, oh, like if an animal got injured, we blood, but I'm like, oh, but it'll probably go bad before you would even collect it.
Yeah, so that's a good question. I don't know the answer to that.
I don't know,
Like
but-
aquatic, like if you were doing that for like a lake or something.
Yeah, you could. So you just take a water sample and
See what
do the send it to the molecular people, and I mean, it could just be like you're just looking in general, but if you're looking for something specific, I think it goes a lot faster.
According to all of my parents' forensic shows that like to watch, DNA can last a long time. So a lot of them were cold cases where they were able to match DNA and able to match that to genealogy, like their family tree.
Use the 23andMe to catch the.
They actually did that a couple times and they were successful. It's pretty neat.
I've heard that. Alright. Let's see, some other stuff about diversity. It's just kind of like random things on each slide. So, microbes and endemism. So for a long time, we all kind of thought that anything that was microscopic was cosmopolitan. You found everything all over the world. And so everything is, in this quote, I don't know who this person was, but everything is everywhere the environment selects. So everything could be found all over the world, but you found different communities and like 2 different lakes that were far apart because the environment selected for those particular species that you find there. But originally, like, all the species could be there, but they just don't last. And now we don't really think that so much anymore. One group of microbes, the ciliates, little dudes with cilia, they do seem pretty cosmopolitan, and we do think that the ones that we find are pretty much all over the world. But when we look at a lot of the other groups of microbes, and I can confirm this for diatoms, there's often some species that you can find all over the world, but then others that are endemic to certain places. You can only find in certain places. And so, and there's kind of a dispersal limitation on microbes. I'm not sure how all the ciliates got dispersed.
Because some of this got dispersed from when, like, say, like the Ice Age and all the ice caps retreated back.
Yeah, I mean, I could move organisms. Also, like waterfowl that migrate, they can carry microbes on them, in their feathers, on their feet, in their digestive tract, and that would be easy to kind of spread it around. So the microbes need help to be dispersed. They're not really going to do it on their own. So we see some things that are more cosmopolitan, but then we also see some endemism and diatoms for sure. There's certain things that we might find only in like one location in Europe or the Rocky Mountains or Alaska, wherever it is. So that's kind of cool. Alright, let's talk about invasive species. Holy moly! We have a lot of non-native invasives that are in aquatic systems. And invasive, non-native species, so non-native does not necessarily mean invasive. And actually invasive could be a native species. So, invasive species are ones that have become so abundant that they cause problems in the ecosystem or in the food webs. They're causing some kind of disruption. And we have a lot of introduced species in aquatic ecosystems. Some of these introductions are intentional, like this was my food source from a lake in my home country, somewhere not on North America, and then when I moved to North America, I brought that organism with me and threw it into a lake or a stream. And that can happen. There's also intentional introduction that's not like, it's not like I want to grow this thing, but aquarium trade, that's how some organisms get introduced, is someone's got something in an aquarium, like carp, like a goldfish, and it gets too big or they move and can't keep it, and so they don't want to kill it, so they just let it go somewhere. It can become invasive that way. We've got accidental introductions. There's a bunch that have been introduced to the Great Lakes through ballast water in ships. So ships need to have a certain amount of weight on them so that they're they're stable in the water. So if they're too light, they'll ride too high, and they'll get to be, if they're too heavy, they'll sink. So if you've got a ship that maybe doesn't have a big load and it's in Europe, it'll take in water from where it is into the hull, and then it will travel somewhere else. Let's say it goes to the Great Lakes area, and then it puts a load on from Great Lakes area, and then it would need to get rid of that ballast water to balance its load again. And so you've got organisms in the water from the Caspian Sea, let's say, and then you just spit it out into the Great Lakes. You've just introduced a lot of potential new organisms that way. And so we recognize now that that's a problem, and they do some work to try to avoid that from happening, but historically we've got a bunch of species that have been introduced that way. And these are just invaders in the North American Great Lakes. We've got some fish, we've got some plants, and the zebra mussel, which is a big stinker in the Great Lakes. And non-native does not always become invasive. Kind of the rule of thumb that I've seen is about 10% of introduced species survive, but only about 10% of those that survive become invasive. So like 1% of your original introduced can become invasive. And one species can be devastating. That's true.
It's like they can also become like naturalized.
Yes, yeah.
It's like the red fox.
The red fox.
Because it was introduced by Europeans by fur trade,
Oh, really?
and then,
I didn't know that.
but it didn't cause it's just kind of like integrated into North American wildlife. Because we had like other foxes, like brown foxes and stuff in North America, but they wanted the orange pelts to hunt, and so they introduced red foxes.
Okay. Interesting. Alright. Another thing about non-native species is you don't often know, like if something gets introduced, you don't often know if it's going to become a problem until it becomes a problem. So all these problems with zebra mussels, I think were not predicted. And most of these invasive species, like the ultimate goal would be to eradicate these species and have them completely gone, but that's really hard to do, and we're not likely for that to happen. Some things we can control, and this might be expensive. One organism we spend a bunch of money to control is the sea lamprey, because that's parasitic on native fish. Do you know about the lamprey? It's one of those early lineages of fish that's jawless, and it just latches on to the outside of a fish, and it has a raspy tongue and just like gives them a big gnarly sore. And so sea lamprey were introduced into the Great Lakes. They were really bad for native lake trout, and we can get rid of the sea lamprey, but they do try to control them. So they've developed lampricides, and so they just try to keep their numbers down enough for the lake trout to be okay. Some more introductions. In general, trout, carp, clams. Water hyacinth is a really bad plant that was introduced in kind of tropical areas. Like there's a Daphnia, so this is a microcrustacean, so there's a few of those. There can be all kinds of weird introduced species. And we could spend all semester just talking about invasive species in aquatic ecosystems. Okay. And a little bit about extinction. So with our old friend human activity, current extinction rates are perhaps 1 million times faster than rates of evolution. So what we're losing, and it's not being replaced very quickly. Yes.
Aren't we the next greatest great extinction event?
Yes. Welcome to the Anthropocene. Yeah. So the extinction. So there's been several mass extinctions in the history of life, and we are in one that we've caused that's happening.
It's like the 6th one or something?
Yeah, and it's like much faster rate than all the other ones. And, you know, given enough time, diversity will come back. We just don't know what it's gonna be, because you never know what mutation's gonna give you, but, we won't be around to see it. And, so that's an issue. Here in the southeast, I don't know if you know this, but, southeastern US is actually kind of a hotbed of, aquatic species richness and diversity. See, I haven't been to this website for a while. Let's see. So, one second. So Center for Biological Diversity, they talk about the Southeast freshwater extinction crisis. So as we, of course, human activity impacting freshwater ecosystems in the Southeast, we lose species, but we have a lot of fish species that are 62% of fish species in the U.S. were found in the Southeast. Mussel species, 91% of U.S. species found in the Southeast. Almost 250 dragonfly and damselfly species also found in the Southeast. So, huge amount of richness in aquatic species in the Southeast. So this is a Halloween darter, because when the males are feeling sexy and they get all colorful for breeding season, they have black and orange stripes on them. Not purple and green, which are just like bastard Halloween colors. They're wrong. It's just wrong. See some other stuff. So other extinction issues, we have been seeing global amphibian declines for the past few decades, and so habitat degradation and destruction, that's kind of an overall gigantic problem for, or reason for a lot of the extinctions that we see. But we were seeing amphibian declines in places that were not disturbed, and it took a while to figure out what was going on. And it turns out there's this fungus, there's a group of fungi called chytrids, which are a very kind of primitive group of fungi, and likely these fungi were spread around the world by human activity. I mean, imagine, you know, you go hiking in the Amazon, you get some mud in your boots, you get on an airplane, you go somewhere else that's thousands of miles away, you put on your boots, and like the chunks of soil that are stuck in your boots get transferred to where you go next, and there could be all kinds of microbes in there. So chytrids got passed around the world, and so places that where the frogs weren't adapted to those started to get these skin infections, and so the fungi would infect the skin of the frogs, and it's eventually a fatal disease. So chytridiomycosis has been causing problems for amphibians around the world. So there's that. So we have pollutants are next. We'll talk about those another time. Any questions? It's kind of a like random slides lecture. Just some days I'm feeling kind of random.